So commensal gut bacteria has immune modulatory effect, and they also provide a baseline homeostasis. And what does this really mean? It means that the commensal bacteria is stimulating the same immune systems which maintain defense against the dangerous bacteria. So commensa maintains mucin production by the goblet cells. Commensal bacteria will maintain and stimulate IgA secreting plasma cells. They will also maintain a continuous production of antimicrobial peptides. And this baseline activation of the mucosal surface and the epithelial cells is very important for the preparedness to defend against pathogenic bacteria and viruses. Okay, so now we're going to talk about the gut and then the lung, and then we're finished. So, just underneath the gut mucosal epithelium, we find the lamina propria. In the lamina propria, there are small lymphoid organs called Peyer's patches, which is shown on the picture. So first, very short about the physiology of the gut. The small intestine and the colon look slightly different. In the small intestines, we have these villi that stand out and increases the active surface of nutrition uptake. In the colon, we don't really have the villi anymore, but we still have the crypts. So now when we talk about the gut, we are normally referring to the small intestine, and there are several different of them, but from an immune perspective, they function similar. So in the villi, there are these intraepithelial lymphocytes, which are mostly ILCs. Underneath, in the lamina propria, we have the dendritic cells, and the dendritic cells can protrude arms of cell protrusions and sample the content of the lumen for antigens. Just in the crypts in the small intestines and the large intestines, there are Paneth cells, which we talked about before. And just above the Paneth cells, which are not shown here, are the stem cells. The gut epithelium is highly proliferative, and as new cells are produced by the stem cells in the crypts, the other cells are pushed outward and forward. And towards the top of the villus, the cells will then shed. And this is also a defense mechanism, since the cells don't get very old, and if there is an infection or bacteria attaching, sooner or later these cells will be shed and disappear. The Peyer's patches are found in the lamina propria, as we saw before. The follicles inside of them are the B cell follicles. The Peyer's patches sample their antigen to be presented to the adaptive immune response through the M cells. On top of the epithelial cell layer, we have the mucus and the goblet cells, and also depicted here to the right in the lamina propria are the plasma cells producing IgA. Depicted on this slide is basically exactly the same thing, but from a different book, because it's important to repeat. So what is different here is that they show the lymph system, the lymphatic system, which is draining towards the mesenteric lymph node. They are also pointing out that there are isolated lymphoid follicles, which means that there are collections of T and B cells and antigen-presenting cells in small follicles, which can be seen in histology, which are totally normal and doesn't constitute infection, and they're smaller than Peyer's patches. So these organs and the Peyer's patches are collectively referred to as GALT, gut-associated lymphoid tissues. So the M cells are very important for providing antigen to the antigen-presenting cells in the epithelium. The M cells are often sitting on top of the Peyer's patches, but they can also be found elsewhere through the epithelium. And they can transport whole microbes or only pieces of microbes and molecules over the barrier. And the resulting antigen is handed off to dendritic cells. So now we will talk about the lung, and that is important in these times. So the lung is normally divided up in the conducting portion, which is trachea and the bronchi, and the respiratory portion, which is the alveoli. The bronchioles, which are small, usually smaller than 2 millimeters in diameter, is the transition between these two parts. In the conducting airways, we have goblet cells, like in the gut, and we have a type called basal cell. In both the bronchioles, the trachea, and the bronchi, we have the ciliated cells. The club cells are only present in the lower part of the conducting airways, and their job is to engulf airborne toxins and break them down, because they have a cytochrome P450 enzyme, which can do that. The alveolar airspace is very special, and we will come back to that, but it has specialized epithelial cells for gas exchange and specialized alveolar macrophages. So we start with the conductive airways. The structure of the conductive airways epithelium is pseudostratified. And of course you remember what that is, because you remember the first pictures. So in here we have the goblet cells. We also have the basal cell, and the basal cells are located below the surface of the epithelium, and they are progenitors of both the ciliated cells and the secretory cells, and they are important for regeneration. So this is a precursor, dividing precursor. So on top of the goblet cells in the conductive airways producing mucus, there are also secretory glands, which is called a submucosal gland, which together will contribute to produce the mucus which keeps the airway lubricated. The cilia on top of the ciliated epithelial cells is flipping upwards in order to remove bacteria and particles that have come down into the lung, and therefore they will end up in the nasal cavity or in the mouth. So here's a closed up picture of a submucosal gland, and in red you see the secreted mucins, and they are produced by the goblet cells situated in the gland. In green we have surfactant, and surfactant is lining the whole lung and preventing collapse, and it is constituted by lipids and proteins that are produced by type 2 epithelial cells. We'll come back to that. The proteins in surfactant is the SPB and SPC, and the lipids have a hydrophilic head group which is sticking in towards the mucus, but lipophilic tails which are hanging out in the air. So in the respiratory airway, which is the alveole, we find two distinct types of epithelial cells. The type 1 epithelial cells, through which the gas exchange occurs. The type 2 epithelial cells was mentioned before. It is producing surfactant. And here is also a picture of the surfactant. It has a very particular three-dimensional structure. And the squamous type 1 alveolar cells cover approximately 90% of the alveolar surface. So the surfactant itself has an antimicrobial activity, and surfactant-associated innate defense molecule can bind and aggregate or directly kill microbial pathogens. This will also enhance their clearance by professional antigen-presenting cells in the immune system and minimize inflammation. The third cell type, which is very important in the alveole, is the alveolar macrophage. The alveolar macrophages are intrinsically anti-inflammatory in order to prevent inflammatory damage to the lung. Another important function of the alveolar macrophage is to recycle and break down the surfactant, which can then be rebuilt by the type 2 epithelial cells. So the immune system in the airways can be activated by allergens, DAMP, which is damage-associated molecular patterns, PAMPs, which is pathogen-associated molecular patterns, or directly by pathogens. The patterns are recognized by the pattern recognition receptors, which we'll talk more about tomorrow, and they are expressed on respiratory epithelial cells as well as on immune cells. The binding of the ligands to these receptors results in an activation of the epithelial cell intrinsic signaling pathway, which is in NF-kappa B mediated. The production of cytokines and chemokines will then in turn activate the immune system further. But it's important that in this organ, the epithelial cells themselves serve as immune cells to a large extent. So when the epithelial cell encounters, for example, the TLR4 ligand LPS, it will activate NF-kappa B via MYD88. On top of that, IRF3 will be activated via a different pathway. Together they will produce cytokines and chemokines, and the IRF3 will produce interferons, which will recruit and activate immune cells, or directly, as in the case of interferon, activate neighboring epithelial cells and help them to restrict viral replication. Type 1 interferon has been shown to be very important in the beginning of the SARS-CoV-2 infection. The immune cells which are then recruited to the epithelial cell layer is depending a little bit on which type of signal is sent out. So the alveolar macrophages are a very specialized type of macrophage, and it is not recruited from blood. Alveolar macrophages maintain their own proliferation using GM-CSF. The alveolar macrophages also have a slightly different surface expression of molecules, which is very confusing because they do not express CD11b, but they do express CD11c. The alveolar macrophages are suppressing DC and T cell responses via TGF-beta and IL-10 production. The alveolar macrophages can be rather seen as a janitor, and not a very aggressive one, but a very soothing and calming janitor. That's a nice guy.